Hello dear viewers, I am Divya, Head of Department in Faculty of Pharmacy in RPIIT campus. I welcome you on RPIIT Academics. Dear viewers, in this video we will discuss about the process of microscopy of stomata. Before going to the experimental part, I will let you know what are the stomata. Stomata are the tiny pores which are present on the leaves. These are present on the upper or lower surface of the leaf. These are the minute pores which are responsible for the exchange of gases. It is responsible for the intake of carbon dioxide and the oxygen is released. It is also helps in the removal of water from the plants. So let's discuss about the stomata by the experiment. Dear students, here we have taken a leaf of Rauwolfia. First of all, we have to clean the leaf with the help of water. Then dry it properly. This is the upper surface of the leaf, that is more greener than the lower surface. This is the lower surface of the leaf. You can see lamina and midrib and veins on the leaf. Next is you have to tear the leaf from the upper side to lower side. After tearing off leaf, you can see a transparent layer which is a epidermal layer. The epidermal layer consists of stomata. Now this is the epidermal layer. It is transparent in color and it consists of stomata. Stomata are numerous in the leaf surface. The next we have to do is, with the help of a sharp blade, we will cut the epidermal layer and place it on the glass slide. Before placing it on the glass slide should be cleaned properly with the help of a cotton cloth. This is the epidermal layer which is transparent in color. The next we have to do is, we will take 3 watch glasses. First is of pure water, second is safranin and the third is also of water. We will put first epidermal layer in the water, then secondly we will shift it in the safranin solution present in the watch glass, and the next we will shift it into the third watch glass which consists of pure water. The reason behind to shift it in the water is to remove the extra staining agent if present. This is how the extra stain of the safranin will be removed from the epidermal layer. The epidermal layer can be stained properly with the help of staining agent, that is safranin. Now with the help of a brush, you can use a paint brush, and the epidermal layer which has been stained with the help of safranin is placed on the glass slide. Now you can use glycerine, just 1 to 2 drops of glycerine. This is added just to make it prevent it from the drying. Afterwards, the glass clip is placed on the glass slide, and very carefully this process is being done with the help of a needle to avoid any bubbles. Now we can see the structure, the epidermal layer under the microscope. The lens used is 45x. This colored minute pores are of stomata. How wonderful they can be seen with the microscope. If we increase the power to 100x, then the structure can be seen this way. You can see number of stomatal cells, and the guard cells are surrounded by and this minute structures are of stomata. The stomata are open this time. You can see number of stomata which are of bean shaped under the microscope. These are the microscopic structure of stomata. Bean shape and it consists of small pores through which exchange of gases can occur and the water can be removed out from the plants. So dear viewers, I hope you like the video and this video might be